If you have the relationship, for example, here if you see the property delta P is related with the delta delta G, you can relate delta delta G equal to some constant plus m into delta P. So, delta P is a property value and C is a constant, because if you make a straight line, right, if you see the values like this, right, we can use this equation to calculate delta delta G, and then you can see whether you can predict. So, if you have some set of values and you frame the equation, and then if you have new mutation, right, you know the C and M and substitute value delta P, you can calculate delta G, right, you can predict stability if you have this a good correlation. For example, if a single property cannot account the stability of this mutant, what can we do? What the other options? You can have many properties. We discussed about 49 properties, right, physical, chemical, energetic and conformational, right. You can use different properties based on multiple regression technique, because you extend these variables. We try to fit these equations with known values using principle of least squares, and we get the constant values, right, for actually m1, m2 and so on, right, you can add this. Once the constant values are known, the coefficients are known, then for any mutant you calculate the properties and use this equation to predict the stability. This is based on multiple regression technique. Then also there are different other techniques such as knowledge based prediction, because you can obtain the information from the mutation and we can use the information for prediction. Then we can use a classification regression tool, the average assignment method, pair potentials as well as sequence based prediction. So, currently in the literature several methods are available for predicting the stability change upon mutation. So, we will discuss the few methods, right, mainly the average assignment methods, pair potentials as well as how to predict from the sequence. So, if you search the Protham data, you will get plenty of data. So, we look into this data, right, several you can see several data you can see the repetition. For example, if you see the alanine to valine for any particular protein, you can see this data can be obtained at different temperature or different pH or different buffers and different concentrations. In this case you can have different data for the same mutations. You have to check carefully, and first we have to remove the duplication of data. It is also possible that same data could be reported from different papers, right, because research is open to everyone. So, not necessarily that only one group should do the particular type of research, because it is very competitive. So, many groups at the same time work on same proteins. In this case, when they get the data, they will publish with their information, they will publish. So, it is possible to have the same data from different groups. In order to accumulate the data, so the Protham considers all the papers published in the literature, because we are not sure which one we need to take. So, in this case we have to remove the duplication in different ways. If it is unique, you can take the data as it is. If it is duplicated, you can check the data, either you read the papers and see the method, which method they use, or see the average values. If the data are very close, for example, say the deviation of less than 1 kilocalorie per mole, right, in this case you can take the average for the particular mutant. If it is very deviated, you can remove the outliers and get these average values, and you can use that values for constructing a model. And you can also try to predict the extreme stability, on what conditions, why it is extreme stability, that also you can try to analyze. So, now then you can also consider secondary structure as well as orbital accessibility, right, you can see this effect. If you want to do this, if the structure is not available, then we can either you can predict or you can discard the data. So, you can both options, one we can predict the secondary structure and ASA, and you can use for prediction, right, or if you do not have, if you need only the structure information, then if it is not there, then you can discard this data. So, finally we can develop the data set. So, if you have the data set, right, with all the data, then we can construct the frequency of mutations. This is the data I show for all the mutations. When you construct your data set by removing all the duplicate data, and if you take only the unique data, so you will get the similar type of matrix, right, with your data set. What is how many data in this matrix? Usually we get
20.
20 into
19.
19, this is equal to 380 mutations. As I discussed earlier, so all the mutations are not uniform. Some cases we have more number of data, some cases we have less number of data, right, depending upon this, right, the mutant type. Mainly alanine mutations we have more number of data, and other mutations, for example glycine to methionine is 0. So, many cases we have less number of data. The average assignment method works based on number of data. So, if we have more number of data, then we will get better results. If we have less number of data, in that case we do not know about the performance of that method for that particular mutant. So, here I show you the frequency of stabilizing and destabilizing mutations. We use a specific particular conditions, for example neutral pH, right. If it is 7, we can take 5 to 9 and particular temperatures, and finally we collected all the data and we classify into two groups, one is stabilizing and one is destabilizing. So, here if you see the numbers, they are given for the destabilizing mutants, and the one in the parenthesis are the one for the stabilizing mutants. If you see this table, we can derive some conclusions. For example, some mutations, for example valine to alanine, right. Where is valine to alanine? Here. Most of the mutants are destabilizing. How many totally how many mutants? 48, right. 48, 44 are destabilizing. That means more than 90%, about 90%, right. Likewise, if you can you find some other mutations which are mainly destabilizing, say W2 alanine, here totally 10 mutants and all the 10 are destabilizing, right, this is also destabilizing. So, any other examples? Phenylalanine to alanine, phenyl F2A, right, yeah, this is F2A. If you take F2A, how many mutations? 27 or 27, 25 are destabilizing, right. Like it's Y2A, this is 23, I2A 37 for 37, I2T 8 by 8. So, from this frequency table you can see that some mutations are always destabilizing or most of the times they are destabilizing. This is unique data that you can get from this Proteome database with some conditions. This way the number of data are less, but you can see a trend. Likewise, if you see the stabilizing mutations, for example, you take N to I. What is the case of N to I? 2 out of 2 by 2, right, this 2 by 2. There are only 2 mutations are stabilizing, or both of them are stabilizing. What is K to S? There is only 1, this is 1 by 1. So, any other stabilizing mutations, if you see here, this is a 2, E to W, right, this out of 2, both are stabilizing. But the numbers are very less because it is very difficult to stabilize a protein compared to destabilizing a protein, because if you make any mutations, they will destabilize a protein. So, because it is very important to enhance the mutations, this way the stabilizing mutations are less. So, this is the reason why the numbers are less. And if you have some cases, they will both stabilize and destabilize. For example, take K to M, right, out of 9, if you take K to M, 9, 4 are destabilizing and 5 by 9 are stabilizing. In this case, we do not know if you change the lysine to methionine, it may stabilize or it may destabilize. So, can you tell some other example which can act as both stabilizing and destabilizing? E to L, what is E to L? E to L 2 comma 2, this is what are 4, 2 are 50%, right, it is just we do not know. So, if you do the average assignment method, if you can see many mutants are destabilizing, for example valine to alanine, if you have a new mutation valine to alanine, you can assign okay this is destabilizing, or if you find a mutation, right, asparagine to isoleucine, you can easily say that okay this will stabilize. On the other hand, in some cases, right, for example V to I, E to K, right, what is the V to I? This has V to I 8 and 10, 8 by 18 and 10 by 18, right. In this case we do not know whether stabilizing or destabilizing. With the maximum number we will take stabilizing, right. In this case we will have a lot of negative values, right. So, we assign these numbers and we can predict the stability change, whether this will increase or this will decrease. It is very easy. Make a matrix. Now, we know the stabilizing mutants and destabilizing mutants, and you can make a table. These are the mutations which stabilize, and these are the mutations destabilize. If it is 0 0, then we do not we cannot predict anything. For any mutation, if we take a protein, each location, it can be 19 possibilities, right. You can do these mutations, right, and get the percentage accuracy with known data. We can get about 60 to 70%, up to 70% accuracy you can get, depending on the data, because in this case you will get the very high performance, because these mutations are very frequently occurring mutations. In this case your accuracy is very high. So, what will happen if the stabilizing and destabilizing effects, even in the stabilizing effects, some cases few of them are in the other hand, right, because this one we considered all the mutants together. Now, it is possible to classify these mutants based on the secondary structure or based on solvent accessibility. If you classify this based on secondary structure, we can make 3 classes: helix, strand and coil. If you classify based on accessibility, then also we can make 2 classes or 3 classes, right, for example buried or partially buried or exposed and exposed, right. You can make 3 groups of secondary structure, 3 group for accessibility, then you have 3 matrix based on secondary structure and 3 matrix based on accessibility, right. Then if the new mutations come, you will check the secondary structure. If this is helix, then pick up from the matrix from the helix. If it is strand, then pick up the matrix from strand. Or you can take this is buried, if from the buried and the exposed from the buried exposed, right. Then you can combine, make 9 matrices. For the helix based on solvent accessibility 3, for the likewise strand and coil 3 multiplied by 3, we can get 9 matrices if you classify based on both secondary structure and ASA. Now, if you have new mutants, first you check what is secondary structure, what is solvent accessibility, then accordingly pick up the data, right. So, what is the disadvantage of having this classification? What is the advantage of having this classification?
More accurate prediction.
Yeah, advantage is okay, because even if the same mutation can stabilize and destabilize, depending on the location you can classify. In this case your accuracy will improve. Disadvantage, if you see, you do not get sufficient number of data, although here it is many zeros. If you classify, then all these 20 will be into 9 groups with less number of data. So, the data is the problem. If you have more number of data, right, then it is easy to classify and you can get better predictions. This is a classification. Now, if you want to predict the real values, right, what can we do? For example, if we have these mutants, for this is C to A, it is 4, all the 4 we know the values, take the average, now this is the value, right, minus 1.9. These for the destabilizing, and for the stabilizing we give the values in the parenthesis, right, we have the values. So, now if we have a new mutation, right, for example it is alanine to valine, so you can say this is minus 1.7, because this is the highest value, so put minus 1.7, then assign all the values and you can calculate the error. Then how to calculate the error? That is experiment minus predicted. You will take the absolute, because either experiment will be higher and the predict will be higher, and you add up all these values and you will be n is the total number of mutants. You take the predicted values, take the absolute values and you can get this n. Also you can calculate the correlation, because we have the experimental values here, the predicted values also here. So, you can this is the experimental, this is the predicted. So, you can get the correlation. Okay, so, what is the status? If you do like this, you can get around up to an accuracy of about 70%. You can discriminate the stabilizing and destabilizing, but if you classify with the secondary structure and ASA, you can increase this up to 80%. Right, this is the, if you make the different tests, then you can get up to 80%. Take the correlation, this is about around 0.5, and here also you can see the correlation about 0.7. So, that means if you make any method, this is the minimum accuracy and correlation you can get, right, just from the assigning the method. Now, you can add more information to increase the performance, right. Let us see what are the other different methods. So, one of the most popular methods, this is based on energy, because energies are very important for the different interactions, and if you mutate a specific residue, this will spoil the energy, it will disturb the energy or improve the energy. This can tell you whether it will increase the stability or decrease the stability. For getting this energetic approach, so what can we do? First we need to know, right, what will happen if a particular residue is mutated to other residues. That means, if you have residue A, how far this is interacting with other residues, how this residue is making contacts, if you mutate, how the contacts will change. Second aspect is, what is the torsion angle for this particular residue, if you change the conformation, how this will change the conformation, how this affects the stability of the mutations. Based on these aspects, first we need to know, we collect a set of proteins, for example around 4000 proteins, they are with high resolution, maybe less than 2 or 2.5 angstrom, and the less sequence identity. Then we classify into different groups, location of each residues based on ASA and the secondary structure, right. This will give you the environment. Then see, then finally we need to do the potentials, how far each residue in each secondary structure or depending on each accessibility, they are making contacts with the different residues. Likewise, you can see the torsion potential, how far the conformation will change, how the conformation will affect the stability. So, once you have these values, we have it for the wild type. When you take the mutation, you replace the residues, so that what will happen if alanine is fell into mutated valine. So, take these tables, take the new values, right, and then you predict the stability that delta G mutant minus delta G wild, because the wild type we know that when you make the mutation changes we know, from that you will calculate the delta G mutant minus wild. Okay, I will explain how to do that. First we classify the atoms into different types. Here we make 50 different types for different amino acids. For example, this is the carbon C alpha. For glycine, only hydrogen is a side chain. So, here we treated this glycine carbon separately. For all the carbons which have the C beta, we take it as 1. Likewise, a nitrogen, all these nitrogens we take a separate type. Likewise, if we have different types of atoms, we treat separately. So, we made into 4 different types of atoms based on location and the connectivity and the chemical nature. So, what is the chemical nature? Where this atom is located and where it is connected, right. So, we have the 40 different types of atom types, 36, 37, 30, 40, right. So, now for all these 40 types of atoms, we can calculate the distance potential using the preference of residue pairs. We can see the G or D, this for any particular distance, ING or any specific residues, right, alanine to valine, alanine to aspartic acid, right. So, what is the preference of having these 2 residues in contact with any specific distance, say for example 6 angstrom or 8 angstrom, and here the same distance totally how many contacts. Using this information you can calculate the torsion potentials in the minus kT logarithmic of this probability to get the torsion potentials. This will give you the distribution of residue pairs INJ at any distance d, right. We can optimize the distance, right, with using different distances. Finally, how this will relate with the experimental data, we can optimize the distance, fine. So, when you get the torsion distance potential is done. So, second aspect, you can see the torsions, right, you can use this similarly, you can use same equation, delta G torsion, this is equal to G of phi and psi for any reference points, and normalize with any reference point, because they use various combinations and see what is the probability of having the particular conformation, right, compared with the all random conformations. Then we convert this into potential by multiplying with minus kT logarithmic of this one, right, you can see torsion potentials. So, 2 different aspects, one is based on distance, one is based on torsion, then we combine these 2, right. So, here you can see the torsion potential and the distance potential, right, and this is a constant, this is a kind of multiple regression technique. For all the cases we know the values, because we know the delta G torsion and delta G the distance potential. Delta G we know, then we use principle of least squares. So, use the least square fit, this will give you this B0, BA and then beta. Once we get these values, then for any data you know the g and g star, substitute the values and you can get delta g. Right, here show the results. For example, if you take the 1538 mutations from Protham database, right, because here you get reliable database. So, you can see the relationship between experimental delta g and the predicted delta g. So, you can see the accuracy about 85%. Accuracy means distinguishing stabilizing and destabilizing mutants, and the correlation is exact relationship between experimental and the predicted values, right, this is 0.86. Now, the question is how this performance varies with respect to secondary structure or with respect to solvent accessibility. So, I show the data. So, we classify the ASA in different ranges 0 to 2, 2 to 30 and so on. So, these numbers also we made a classification such a way that the data can be related with the stability with the highest performance. So, accuracy it is about 90% if it is buried. Even in the case of beta strand, if it is buried, then you can see more than 90%. We discussed earlier with the buried mutations you can easily relate with the hydrophobicity and single property can also explain the stability, but in the case of exposed, right, single property is very difficult to explain. This is the reason why we include the sequence information or structure information. Even if you add this structure information, the performance is only limited. So, we see here the 60%, 60 plus solvent accessibility in helix, the performance is about 80% and the correlation is around 0.85 and so on. So, for the buried one you can see it is higher. This for the sheet, the buried one you can higher performance, right, here also you can see. And importantly, if you notice here that the partially exposed case or partially buried case, okay, these values are less compared with the completely buried or completely exposed cases. Now, you can have a server, right, this is the CUPSAT server, right, CUPSAT, yeah. She takes the PDB ID, right, and you have to give the residue number and the which is the residue, either you need thermal or denaturant, right. If you predict stability, first it will automatically calculate the secondary structure and accessibility, it will calculate, torsion angle also it will give you. If there is any problem in the torsions, it will tell you the torsion is a problem. In this case, the results are not reliable. If it is fine, then we proceed to the next step. Then finally, here it will give this is the prediction results. For this mutation, for this isolation is mutated to all the 19 cases, and here the overall stability was stabilizing or destabilizing, torsions favorable, non favorable, and you can see the delta predicted delta G. So, one stretch you can get all the values for all the 19 mutations. This case it requires the structure, because we need to calculate the distance potential and the torsion potentials, that requires structure. Based on that, they check the available database and then see how they are attributed in terms of distance and torsion potentials, and we can get the delta G values. So, the structure is not available, then what to do? There are two different ways. One is you can do the modeling to get the structure and use the server, or we are trying to get a new method just from sequence information, right, because in the average assignment method I mentioned that if this mutation is aligned to valine, it can be stabilizing or destabilizing, right, depending upon the values. Then the sequence information we can tell if aligned to valine and the neighboring residue is aspartic acid, and then this could be a stabilizing residues. So, you can make rules, right. In this case you can take all the information, wild type residue, mutation residues and neighboring residue information, right, all this information we take that, and then we can develop the rules. For example, wild type residue is alanine, right. So, A is the wild type residue, A is mutated to any other residue, and if the neighboring residue contains glutamic acid, then this is destabilizing, right. In this case you use various options available. So, it could this rule can predict up to the accuracy of 97%. Second condition, with the wild type residue glutamic acid, this mutated to other residues, and the second neighbor, this is the N terminal, this is the C terminal, right. Second neighbor is this EM, then this will stabilize the protein, and we have set of data. So, we could get an accuracy of 100%. So, here the problem is if we have more number of mutations and we could accommodate all the mutations with less number of rules, we can get better accuracy, right. So, we consider about 3000 mutations and made about 100 rules, but that means approximately 30 mutations. Some case we have 100, sometimes we have less. If your data are very high, like the well into alanine mutations, right, then we can predict with high confidence. So, we have the server, right, this can get an accuracy of 82% and the correlation of 0.7. So, this is server, it will take the mutation residue and this wild type and the mutation residue, right. Okay, this is the wild type, this is mutant, right. So, we have neighboring residues. Now, we can if we click predict, this will tell you the whether it is stabilizing or destabilizing, right. This will tell you this is the stability change, this is the stabilizing or destabilizing, right, and this already gives some information how many hydrophobic residues nearby based on this mutation and so on. So, one example I give T225U, right, here the experimental TM is 33 degrees and our prediction is also minus 0.97, both are destabilizing, but here we know that only the TM values, but that is anyway higher than 2 to 3 times higher than the delta G values. So, you can use the server also if you the structure is not available, right, you can use server for predicting the stability upon mutations. So, summarizing, so what did we discuss today? Stability upon mutation, right. If we have which database we use? Protham. So, if data is available, first you can try to relate the amino acid properties with stability. Stability values you can get from delta G or delta Tm. Properties, which are various properties? Physical, chemical, energetic, conformation properties. So, you can relate with the correlation coefficient, right, because delta G we know delta G mutant minus delta G wild. Properties we know delta B mutant minus delta B wild. For all mutations you can relate with the correlation. So, this will tell you which properties are important for explaining which type of mutants. Then we can develop different methods either from regression techniques or average assignment method or different potentials, right, to predict the stability. Well, also you can use different rules for predicting the stability of these mutations. So, we derived various structure based parameters, sequence based parameters, and these parameters have potential applications, right, because we discussed about regression techniques, right. So, we can use all these parameters for predicting the stability of mutants as well as other potential applications, for example, to understand the folding rates or to understand how the proteins interact with other molecules and so on. So, that I will explain in the subsequent classes. Thanks for your attention.